Point mutations. A point mutation is a mutation that only changes one base. There are 4 different types of point mutations: missense, nonsense, silent, and read-through, and it is important to understand the differences. The strain of DNA given reads from 5' to 3', ATG, TGG, AAC, GCT, TGC, TGA, CCC, GGC. Every piece of DNA has a coding strand and a template strand. The template strand will tell which complementary bases should be used, yet this is the coding strand. It will give the same sequence of bases in the mRNA, other than the exception that all thymines will become uracils as it is RNA. The mRNA made from this DNA would read AUG, UGG, AAC, GCU, UGC, UGA, CCC, GGC, from 5' to 3', coding for the amino acids MET, TRIP, ASN, ALA, cis, stop, leaving the last 2 codons to not be a part of the amino acid sequence. Based on this piece of DNA, we can explore the different point mutations, starting with a missense. A missense occurs when the change in base results in a changed amino acid. If we were to switch the C to a T in the 4th codon of the DNA, that would change the mRNA codon from GCU to GUU, which would change the amino acid from ALA to VAL. This would be a transition, as the base would change from a pyrimidine to another pyrimidine as we changed a C to a T. A transition is a change to the same type of base, such as a purine to a purine or a pyrimidine to a pyrimidine, while a transversion changes the type of base, such as a purine to a pyrimidine or a pyrimidine to a purine. The next type of point mutation we will explore is nonsense. A nonsense occurs when the change in a single base results in changing an amino acid coding codon to a stop codon. If we were to switch the C to an A and the 5th codon of DNA, that would change the mRNA from TGC to TGA. This would change the amino acid from cis to a stop codon, signaling the cell to stop translation early. This would be an example of a transversion, as we would change the base from a pyrimidine to a purine as a C was replaced with an A. Another important point mutation is a silent mutation. This occurs when a base change results in coding for the same amino acid. If we were to change the T in the 4th DNA codon to an A, that would change the mRNA codon from GCU to GCA. This would result in both coding for the ALA amino acid, not changing the function. The change from a T to an A would also be another transversion, as it is changing the base from a pyrimidine to a purine. The last type of point mutation we will discuss is a read-through. A read-through is when the mutation causes a change from a stop codon to amino acid coding codon that will keep going and read through where it should end, making the sequence longer. If we were to change the G in the 6th DNA codon to a C, this would result in the mRNA changing from a stop codon to coding for SER. This leaves the last 2 codons that were originally not being used to code for amino acids to be used to give PRO and GLY, making the amino acid sequence longer. The change from G to C would also be a transversion, as it is changing from a purine to a pyrimidine. As we can see, each type of point mutation has a different effect on the amino acid sequence, and that's why it is so important to understand the difference.